Welcome to Labster Simulation. In this experiment, you will discover the function of glucagon, its structure, and its interaction with a glucagon receptor using a bioinformatics website called the Protein Data Bank. Will you be able to know your way around the Protein Data Bank?
A feature that is essential for the life of higher vertebrates is their ability to maintain a constant blood glucose concentration. Glucose is an essential energy source for all cells, especially for the brain and its neurons, which are heavily dependent upon a continuous supply of delivered glucose. However, changes in blood glucose levels may occur as a consequence of physiological and metabolic events. Thus, glucose concentrations in the blood must be regulated.
The pancreas is the endocrine gland that is largely responsible for the maintenance of blood glucose levels, as well as the cellular uptake and exchange of fuel metabolites. The two principal hormones secreted by the pancreas are insulin and glucagon. Which hormone do you think is responsible for elevating blood glucose levels? Correct. Glucagon stimulates the catabolic reactions that lead to the breakdown of glycogen, proteins, and fat, which result in an elevation in blood glucose levels. This hormone acts as a counterbalancing hormone to insulin, which, on the other hand, acts to decrease blood glucose concentration.
To increase blood glucose concentration, glucagon activates several metabolic pathways. What are these pathways? That's right. Glucagon activates gluconeogenesis and glycogenolysis to increase blood glucose concentration, while insulin activates glycolysis and citric acid cycle. The pancreas continuously adjusts the relative amounts of glucagon and insulin secreted in response to the continuous changes of blood glucose and other fuel metabolites, occurring as a consequence of changes in the relative amounts of anabolism and catabolism in various tissues.
The blood concentration of glucose must be kept within what range? Wow, you are so right! The normal blood glucose concentration range is between 45 to 55 milligrams per 100 ml. A reduction in blood glucose levels below this range for a continued interval of time will lead to an impairment of brain function, tremors, and convulsions due to activation of the sympathetic nervous system, which may result in untimely death. On the other hand, a prolonged elevation of blood glucose above 130 mg per 100 ml generates a state of hyperglycemia due to the lack of insulin. You got it! You are starting to become a glucagon expert already. To learn more on how glucagon is synthesized and broken down, go to the Workbench 2.
Found in the islet of Langerhans, which of the following produces glucagon? You are amazing! Produced by pancreatic alpha cells found in the islet of Langerhans, glucagon is initially synthesized as a proglucagon in the rough endoplasmic reticulum. After being cleaved and modified, glucagon will be released into a vesicle from the rough ER, which will then travel to the Golgi apparatus for further modification and packing. Multiple vesicles containing glucagon will then be released into the cell. Since there are low glucose levels in the blood, this means that there is also a low membrane polarization inside the cell, meaning that calcium channels will open and cause calcium ions to enter the cell. This will then bind with the vesicles containing glucagon and help it fuse with the cell membrane, allowing the glucagon to be released into the bloodstream.
Where does glucagon stimulate the breakdown of glycogen? You are very good at this. Once glucagon is released into the bloodstream, this will then trigger glycogenolysis in the liver. This allows the stored glycogen to convert into glucose. Once that's done, glucose will be released into the bloodstream, keeping the blood glucose levels balanced.
Insulin and glucagon are both synthesized in the pancreatic cell. Which among the choices do you think prevents the release of glucagon from pancreas? Correct. Glucagon is synthesized in the pancreas by pancreatic alpha cells. During periods of fed state, the glucose molecules in the blood increases, releasing the insulin from the pancreas. The presence of insulin inhibits the release of glucagon from pancreatic alpha cells.
When insulin is released from the pancreas, which pathways do you think are inhibited in the body? Wow, you are on fire! Since insulin prevents the release of glucagon, pathways that are activated by glucagon such as gluconeogenesis or glycogenolysis are inhibited.
In what situation do you think glucagon is synthesized and released from the pancreatic alpha cells? That's right. It seems like you've been studying. Glucagon is synthesized and released from the pancreatic alpha cells when there are low blood glucose levels. This happens during the fasted state, that is, the body needs to activate glycogenolysis and gluconeogenesis to synthesize glucose molecules. When glucagon is released, one of the organs that it can enter is the liver that contains glucagon receptors. When glucagon attaches to the glucagon receptor in the liver, it will activate the glucogenolysis and gluconeogenesis in the liver, allowing the liver to synthesize glucose and disseminate it to tissues or organs that need glucose.
What is the structure of glucagon? Wow, you're an alpha in this! Glucagon consists of 29 amino acid residues. It has an alpha helix structure with side chains of the same polarity every 3 to 4 residues. Additionally, the amino acid sequence of glucagon is shown below.
To what receptor does glucagon bind? Good job! Glucagon binds to the G-protein coupled receptor in the plasma membrane. The G-protein coupled receptor is also known as a 7-transmembrane receptor that binds extracellular substances and gives signals to G-protein. Conformational change will occur which activates the G-coupled proteins that increase cyclic AMP levels due to the activation of adenylate cyclase, protein kinase A, and cyclic AMP response element binding protein.
Proteins such as glucagon indeed play various biochemical roles in our body. While glucagon specifically assumes a regulatory function or metabolism, the molecular functions of other proteins also involve cellular processes, DNA replication or modification, transcription or translation, intracellular signaling, protein folding or degradation, transport, skeletal or structural, and defense and immunity. Because of this, it is important to study proteins, their composition, dynamics, and structure-function relationships. Through understanding the folding mechanisms and assembly of proteins, as well as their interactions with other molecules, we are able to uncover scientific breakthroughs.
With the advancement of technology, the biological sequence and structures of proteins have been studied. The data collected from these studies have grown exponentially and are stored in databases or databanks accessible by the public. The need for fast and reliable ways to retrieve and understand these biological sequences and structural data have led to the emergence of bioinformatics. Now that we have sufficient knowledge on glucagon, let's now move to Workbench 3.
Which of the following is a subdiscipline of biology and computer science concerned with the acquisition, storage, analysis, and dissemination of biological data? Amazing! You were so smart! Bioinformatics is a subdiscipline of biology and computer science concerned with the acquisition, storage, analysis, and dissemination of biological data, most often DNA and amino acid sequences. Bioinformatics uses computer programs for a variety of applications, including determining gene and protein functions, establishing evolutionary relationships, and predicting the three-dimensional shapes of proteins.
Bioinformatics tools such as GenBank, NCBI, and the RCSB-PTB are the most widely used in studying biological data. In this experiment, we want to explore more on the amino acid sequences and the structural characteristics of the hormone glucagon, which is the most suitable bioinformatic tool to get such information. Wow, you're a pro at this already! The Research Collaboratory for Structural Bioinformatics Protein Data Bank provides an information web resource to biological macromolecular structures. It includes tools and resources that can be used to understand the relationship between sequence, structure, and function of biological macromolecules. Open the computer to explore more on the Protein Data Bank.
First, search the PDB ID of glucagon. PDB ID stands for the PDB identification code. Every molecular model in the protein databank has a unique identification code. For the protein glucagon, the PDB ID is 1GCN. Type 1 GCN on the search box.
Once you search for the PDB ID of glucagon, various data regarding its structure, its macromolecule content, and literature regarding the hormone will appear. First, let us explore the amino acid sequence and structure of glucagon. Click on the 3D View Structure button to get a closer view of the structure.
The 3D structure of glucagon is shown on the screen. You can manipulate the structure by dragging your mouse and rotating the molecule. The computer software offers various tools, and viewing the molecules will be able to study its structural properties. Different models, commonly used in studying proteins, include the ball and stick model and cartoon model.
What type of model is used as shown in the image? You're so good at this! Cartoon model emphasizes the position of the main chain and the associated secondary structures of a protein. As such, this model allows us to easily identify helices and sheets and how they are packed together. In the 3D structure of glucagon, we can see that it is an alpha helical structure. Explore the 3D view and try to change the model from a cartoon model to a ball and stick model. This can be done by clicking on the Actions icon. Click on the Add Representation tab and click on the ball and stick and non-covalent interactions. After this, hide the cartoon model component by clicking on its eye icon. At this point, you will see the ball and stick model of the glucagon.
What do you think is the advantage of the ball and stick model over the cartoon model? Awesome! When you zoom in on the ball and stick method, you will be able to determine specific non-covalent interactions within the protein. You can click on the specific non-covalent interactions designated by the broken lines.
As seen here in the figure, there is a hydrogen bonding between histidine 1 and glycine 4 of the glucagon protein. You can do this with all of the non-covalent interactions within the protein to determine which interactions stabilize the folded structure of glucagon.
One of the primary aspects that influence protein folding is hydrophobicity. When proteins interact with water, hydrophobic residues aggregate within the core of the molecule, while polar residues occur at the surface of the protein. The polarity and hydrophobicity of the amino acid residues in the protein influences the secondary structure that it assumes, and thus, the amino acid sequence of glucagon can also be used to predict its secondary structure. Let us explore the amino acid sequence of glucagon in the protein data bank. Click on the Sequence tab.
As we can see, the amino acid sequence of glucagon is the following. Which part of the amino acid sequence assumes an alpha helix structure? Correct. The amino acid sequence that assumes an alpha helix structure is the following, which resides in the middle part of the protein. You can see this by looking at the secondary structure. That is, the amino acids within the pink line are part of the alpha helix structure. On the other hand, the two ends of glucagon are irregularly structured loops. The amino acid composition and physical properties of a hormone also influences how it interacts with its receptor protein.
Let us explore the structure of the glucagon receptor in a complex with a glucagon analog with a PDB ID of 5YQZ. The image on the screen shows the cartoon model of the glucagon receptor and its analog. The green structures represent the receptor, while the orange structure represents the hormone glucagon. While glucagon assumes a secondary structure characterized by hydrogen bonding, the glucagon receptor possesses a high-order quaternary structure, as its various subunits associate with each other via hydrophobic interactions. As seen on the model, the receptor also assumes alpha helical structures.
The hydrophobic glucagon residues 22 to 27 residing on its near carboxy terminal region are essential for its binding to the receptor. Moreover, hydrogen bonding also exists between glucagon and its receptor. Click on the amino acid residue 28 to view its interactions.
In the image, threonine 28 in glucagon interacts with lysine 41 in the glucagon receptor. Moreover, interactions between the following residues also exist: leucine 26 in glucagon and arginine 93 in the glucagon receptor via hydrogen bonding, and glycine 2 in glucagon and tyrosine 126 in the glucagon receptor via hydrogen bonding. There are many more interactions between the glucagon and its receptor. You can click on the other amino acid residues of glucagon and determine their interactions with the receptor if there are any.
Congratulations! You have successfully completed the experiment. You know your way around navigating through the protein data bank. Through this experiment, you learned about the structure and function of the hormone glucagon as well as how its protein receptor interactions occur. Thank you, and we hope you feel wiser now.